Hello all the students of BIPC first year AP Telangana board. So here we are going to look into short answer type of questions. So far we have dealt completely with LAQs and SAQs till half. Today we are going to look into two major chapters. One is sexual reproduction of flowering plants. Second will be cell the unit of life. Right Nama? So we will get started with the first question. Clear, right? So here if you see, first question says, discuss the various types of pollen tube entry into the ovary with the help of diagrams. Different modes of pollen tube entry into the ovary like porogamy, mesogamy, chalezogamy is not just important for IPE exam, it is also important for your MCET and NEET with the examples. Okay? So when you are going through this question, highlight the examples and learn parallelly. Okay? So here if you see types of pollen tube entry into the ovule. After germination on the stigma, the pollen tube passes through the style and it enters the ovule by one of the following three methods, any one of the following three methods, and mostly it is through porogamy only. First I will explain you the mode of entry by taking the diagrams and then we will go with the theory. So if we see here, first is porogamy. Porogamy is entry of pollen tube from micropylar pore. Okay, entry of pollen tube from micropylar pore is porogamy. Second, mesogamy. If the pollen tube is entering the ovule through integuments, that is called as what? Mesogamy. Mesogamy means entering the pollen tube entry into the ovule through integuments. Next, if pollen tube is entering through the chalazal end, just a second ma. If pollen tube entry is through chalazal end, that is known as chalezogamy. Fine, right? So now we will look into the theory by taking the examples as well. So here if you see ma, porogamy, pollen tube entry through micropyle of the ovule. Pollen tube entry is through what? Micropyle of the ovule. And this is the most common method. Okay, this is the most common method of pollen tube entry, and best example you can take is Othelia or Oinothera. Next is chalezogamy, where pollen tube entry is through what? Chalazal end, which is opposite to the micropylar end or the region of antipodals. This also you can mention, okay, or at the region of antipodals. Example you can take is Casuarina. Next is mesogamy, pollen tube penetrates the ovule through integuments or middle region. Okay, example you can take is cucurbits. Cucurbits is the example. So here this is porogamy, chalezogamy and mesogamy. And then if you see next question, that is on apomixis. Apomixis is the special mode of reproduction. This is very important, not just for IPE again. This is most important for NEET and MCET as well. You can see quite a times the questions have appeared on the apomixis. So here if you look into apomixis is a formation of seeds without meiosis or fertilization, and the seeds which are formed as such are addressed as what? Apomictic seeds. Apomictic seeds. Seeds formed without meiosis or fertilization, and it is a form of asexual reproduction. This is a form of asexual reproduction which mimics the sexual reproduction. Okay, this mimics sexual reproduction. Fine, right? This is seen in some members of Asteraceae and grasses as well. This is seen in some members of Asteraceae and grasses. If you look into the key features, seeds formed without reductional division, that is without meiosis or gametic fusion. Seeds are formed without meiosis or gametic fusion. In some plants, a diploid egg cell develops directly into an embryo to a diploid embryo. Okay, diploid egg cell will develop into a diploid embryo. This will ensure reproduction even in the absence of pollinators. This will ensure what? Reproduction even in the absence of pollinators. That is one major advantage of apomixis. Next is importance of apomixis. What is the importance of apomixis? Apomixis will fix desirable traits. For example, maternal genes are only transported here, right? Without fusion, only maternal genes like egg. Diploid egg is maturing to diploid embryo, right? Diploid egg is maturing to diploid embryo without fusion, right? So, all maternal genes only will be there. This is maternal pattern of inheritance. So, here if you see, what is the major importance of apomixis? If there are any desirable features for this maternal plant, okay, all the desirable features of the parent plant are given to the progeny in this process. So, fixes desirable traits, seeds produced without segregation, okay, directly whatever is there from the parent, all the desirable characters will be inherited to offspring. So, here seeds are produced without segregation. So, superior traits will remain unchanged here. Okay, superior traits will remain unchanged. Next, this will maintain genetic stability. Genetic stability is maintained. Desirable characters can be preserved for many generations. And agricultural advantage. What is the agricultural advantage over here? Extremely useful. This is extremely useful for large scale commercial hybrid seed production, and this reduces cost and dependency on repeated hybridization. This will reduce the cost and dependency on repeated hybridization. Next, if you see it, write briefly about different types of ovules. Okay, different types of ovules. So, here you see we have 6 types of ovules. First, I will recommend, quite a times the question has appeared like in March 2019. So, I will recommend first to draw the diagrams, all the 6 types of ovules, and then explain it. Rather than explaining one by one ovule by the other, you just give out a tabulated form with examples. That will be more advisable. So, here you see types of ovules. First is orthotropous ovule or atropous ovule. Orthotropous or atropous ovule, which is straight ovule where micropyle, chalaza and funicle. See, micropyle, chalaza and funicle will be on the same vertical line. Okay, this is simplest type of the ovule. Simplest type of the ovule. Best example I can give you is polygonum. I can show you the example as well. Here you can see funicle, chalaza and micropyle. They are at the same straight line. Okay, they are on the straight line. This is atropous or atropous or orthotropous type of ovule. Second is anatropous ovule. Anatropous ovule is most common type of ovule. Here the ovule is inverted. Okay, inverted ovule where micropyle will lie close to the funicle. Fine, right? Here micropyle is close to the funicle, close to the stalk. Fine, right? So, here most common type of the ovule in most of the angiosperm where micropyle is close to the funicle and body is inverted to how many degrees beta? 180 degrees. Body is inverted to 180 degrees. Examples you can see is sunflower and brassica. These are the examples of what? Anatropous ovule. Third is camphilotropous ovule. Camphilotropous ovule where body placed at right angle to the funicle. Here body of the ovule is placed at right angle to the funicle, not that one. Here you see camphilotropous, body of the ovule is placed at right angle to the funicle and embryo sac is slightly curved. Here you can see embryo sac is slightly curved. Example you can take is B or Fabaceae. Next is amphitropous. Ovule is completely curved and ovary and embryo sac is into horseshoe shaped. Here camphilotropous, embryo sac is slightly curved, whereas in amphitropous embryo sac is curved to what? Horseshoe shaped. This is amphitropous. Example is alisma. Next is cercinotropous. Cercinotropous ovule where the body of ovule is completely revolved to 360 degrees. Okay, this is initially orthotropous. Later funicle coils around the ovule and micropyle is close to the funicle. Example, best example I can give you is opuntia. Next is hemitropous. Hemitropous you can also address it as hemianatropous. Here body of the ovule is transverse where micropyle and chalaza are into one end. Here you can see hemitropous, hemianatropous where micropyle and here you can see right funicle, body of the ovule is transfer where micropyle and chalaza are perpendicular to the funicle. If this is the funicle, micropyle and chalaza are perpendicular to this. Example is ranunculus. So first I recommend you to draw the diagrams. Okay, I can give you one trick. First you take atropus, then inverted ovule, that is anatropus, and directly you take sarcinotropus, which is 360 degrees rotated. So first atropus, anatropus, sarcinotropus, these three you draw first. In the next thing, first you take hemianatropus, which is perpendicular to each other. Then you take camphilotropus, where embryo sac is slightly curved, and then you take the embryo sac which is curved to horseshoe shaped. These three you draw down and these three you draw above. As such, you remember all the 6 types. First is atropus, anatropus, sarcinotropus, followed by hemianatropus, camphilotropus and amphitropus. Just look at these 6 types of ovules with the examples and you can easily go through this question. Okay, where if you are presenting the diagrams nicely with the examples, you will get impressive marks for this question, because this is diagram based answer. Second important point I would like to convey about this question is, this is very important concept. Types of ovules is important concept for your MSET as well. So likewise you go through this question. Here we go with next question. List 3 strategies that a bisexual chasmogamous flowers can evolve to prevent self-pollination. So what will be the evolution of bisexual chasmogamous flower to prevent autogamy or to prevent self-pollination? Now we have to highlight what? Outbreeding devices. Outbreeding devices, which is important concept for your NEET as well. Fine, right? Outbreeding devices. So here we go. The 3 main strategies used by bisexual chasmogamous flower to prevent self-pollination. What are the 3 main strategies? First is what? Dichogamy. Dichogamy is nothing but maturation of male or female sex organs at different times. At different times they are maturing. If male reproductive structure is maturing earlier than females, that is, stamens are maturing before the carpels. Stamens mature before the carpels, that is addressed as protandry. What is protogyny? Carpels mature before the stamens. Carpels will mature before the stamens, that is protogyny. Protandry, stamens mature before the carpels. Examples you can take like sunflower is the best example. Protogyny, carpels will mature before the stamens. Examples you can take datura and solanum. Next you see hercogamy. This is physical separation of stamens and stigma within same flower. This is nothing but spatial separation. Okay, spatial separation. Spatial separation of stamens and stigma within the same flower. If you see the content to this, anthers and stigma are placed at different heights or at different directions. Best example you can take is hibiscus and gloriosa. Next is what ma? Heterostyle. Here presence of styles of different lengths in a flower of same species. Here styles will be of different lengths. Okay. Presence of styles at different lengths in the same species. Fine, right? Presence of styles of different lengths in same species. This will prevent pollen from falling on the stigma of the same flower. Example you can take is primula, which is diheterostyle. Example is primula, which is diheterostyle. So here we have dichogamy, hercogamy and heterostyle. Clear? Remember it with examples and don't forget to draw the diagrams for this, very important. Fine, right? For heterostyle especially mention, like see at one position the gynoecium is taller than the androecium. On the other hand anther is taller than the gynoecium. Okay, fine, right. Next you look into the next chapter. So from sexual reproduction of flowering plants, what are the concepts we have covered ma for SAQs, short answer type? First is pollen tube entry, where we have to highlight porogamy, mesogamy and chelizogamy. Second question is on apomixis, and third most important is types of ovules. Fine, right. Fourth is about outbreeding devices. These are the 4 concepts where you can highlight for your short answer type preparation from sexual reproduction of flowering plants. Now, cell the unit of life. Cell the unit of life you go through. See, describe the cell organelles which contains chlorophyll pigments. Cell organelles which contains chlorophyll pigments. Many a times asked question, like in 2023 you can see 2022, 22, 21 and 16 as such, most important question. So first, the cell organelle which contain chlorophyll pigment is what? Chloroplast. Chloroplast is the cell organelle which contains chlorophyll pigments. So here chloroplasts are green plastids that contains chlorophyll and they will carry out photosynthesis. This will occur in all green parts of the plant. Okay, this occurs in all green parts of the plants, especially mesophyll cells of the leaves. Fine, right? This occurs in mesophyll cells of the leaves and in green stems. This is usually lens, oval, discoid or ribbon like shape. The size may vary from 4 to 10 micrometers and long and 1 to 3 micrometers thick. Okay, this varies within different species. How many number of chloroplasts can present in a single mesophyll cell? In a single mesophyll cell, how many chloroplasts can present? It can be from few to 100. Okay, it can be from few to 100. Fine, right? So here you have to mention this properly. What will be the shape and how many number approximately per mesophyll cell? And here also I will recommend first to draw the diagram and then go for the explanation. Okay, so started with outer membrane. Outer membrane and outer, it has double membrane bound, or it is a double membrane bound organelle. Outer and inner membrane, where the space between outer and inner membrane is addressed as periplastidial space. What is the space between outer and inner membrane ma? Periplastidial space. Fine, right? And the envelope will enclose a matrix. The matrix of chloroplast is stroma. What is stroma ma? Matrix of chloroplast. Matrix of chloroplast is stroma. And a third membrane system, that is thylakoid. Thylakoid are sac-like structures. Membranous sac-like structures present in the stroma is thylakoid. Okay, these are membranous sac-like structures. And inside the membrane of the thylakoid we have thylakoid lumen. Fine, right? Thylakoid lumen. Fine. Next, stroma is what? This is the fluid filled matrix which is present in the chloroplast. This contains Kelvin cycle enzymes. Those are dark reaction enzymes. Best example is the ribisco. Starch granules, plastoglobuli and ions are present. What is the genetic material here? Genetic material inside the chloroplast is double stranded circular DNA is present and it has 70S ribosomes, same as that of prokaryotic cell. 70S ribosomes, same as that of prokaryotic cell. So the 70S ribosome characteristic feature and double stranded circular DNA, these features can say that chloroplast is evolved from a prokaryotic cell, which is learnt in detail under endosymbiotic theory. Endosymbiotic theory. It will be very impressive if you can mention this term, endosymbiotic theory, over here. Next, thylakoids. Flattened sacs, they are arranged into stalks. Stalks of the sac-like structure is grana. Stalk of sac-like structure is grana. Next, connections. Grana are interconnected and one grana, one stalk of sac-like structures, one grana is connected to other grana by stroma lamellae, or you can say stroma thylakoids. Okay. Now we will go through pigments. Structural description is done. How you will elaborate the structure of chloroplast? First start from the limiting boundary. Outermost limiting boundary that has periplastidal space, then enter into the fluid filled matrix that is inside the chloroplast. Highlight the sac like structures, that is thylakoid, which are membrane bound. You can use this term third membrane system of chloroplast, and then you highlight on the double stranded circular DNA and then 70S ribosomes which are present in the matrix of the chloroplast. Highlight also about stroma lamellae. Now about pigments and photosystems. Thylakoid membranes have chlorophyll A, chlorophyll B and carotenoids. Carotenoids include xanthophylls and carotenes. These pigments are organized into two photosystems. Remember, photosystems are light harvesting complexes. What are these photosystems, beta? Light harvesting complexes. Light harvesting complexes. They will have two set of pigments. One is core pigment. Second one is accessory or antenna pigments. Accessory or antenna pigments. Here you see sites of reactions. Light reactions include what? Photolysis of water, where oxygen is evolved into the inner membrane of thylakoid towards thylakoid lumen. Remember that. Next is electron transport system, which occurs in the thylakoid membrane. Proton gradient occurs in the thylakoid membranes only. Next, ATP synthase will face towards stroma. So, in a single paragraph you have mentioned complete reactions along with their locations. Now, dark reactions as well. Dark reactions or carbon fixation, like Kelvin cycle. Where does this occur, ma? This occurs inside the stroma. Clear till here? Next is genetic or semi-autonomous nature. We know that chloroplast and mitochondria are addressed as semi-autonomous organelles or self-duplicating organelles. Okay, you cannot finish this answer without mentioning those terms, because it is a characteristic feature of these two organelles. Own DNA and 70S ribosomes, these are semi-autonomous and they will replicate by what? Binary fission. Many chloroplast proteins are what? Nuclear encoded or imported evidence for what? Endosymbiotic origin. And this is the structural description of chloroplast. Now we will highlight the functions of the chloroplast, which is very important. What is the function of the chloroplast? First, photosynthesis. If you see, photosynthesis. Chloroplast will trap solar energy. Solar energy is trapped using chlorophyll and they will convert it into chemical energy. Chemical energy here is also addressed as assimilatory power. Assimilatory power, which includes ATP and NADPH. Later it is used to fix carbon dioxide into glucose. Fine, right? Next is food and starch storage. They synthesize carbohydrates during photosynthesis. They will synthesize carbohydrates during photosynthesis, and temporarily they will store them as what? Starch grains. Temporarily they will store them as starch grains. Then is semi-autonomous metabolism. Okay, semi-autonomous metabolism here. Chloroplast will synthesize some proteins, fatty acids, amino acids and pigments. They will synthesize all this. They also contain their own DNA and 70S ribosomes. They will contain their own DNA and 70S ribosomes for limited protein synthesis. This is what, ma? This is for limited protein synthesis. These are the functions of chloroplast. So, what you have to mention about the organelle? First, I always recommend you to draw the diagram, because that stands as a good reference to elaborate the answer. So, first clearly mention about the diagram of the chloroplast with a neat labeling, followed by structural description of the chloroplast, followed by functions of the organelle. This is how you have to finish this answer. Next is What are nucleosomes? Why are they made of? Very important model, nucleosome model, where it has appeared like in May 2017, like March 2014, quite a times asked question, and I always recommend you to draw the diagram first. So, now first I will take the reference of this diagram, I will explain you in detail, then we will go to theory. What is this nucleosome model? From where is this theory came? All the NEET aspirants, you will learn this in molecular basis of inheritance. Of course, in our text it is provided here. Chromatin material, after its duplication in the S phase, where DNA gets duplicated, 2 meter long DNA has to fit in a very small space, that is nucleus, right. So, here chromatin should get untangled to chromosomes, that is chromatin condensation have to happen. In the process of condensation, as DNA is negatively charged, this DNA has to condense. When will this DNA get condensed? This condenses by alignment of positively charged histone proteins. Okay. The negatively charged DNA will get condensed by positively charged histone proteins. What are the histone proteins that are present and how histone protein is positively charged? Histone proteins are positively charged because histone proteins consist of basic amino acids like lysine and arginine. Okay. It consists of basic amino acids like lysine and arginine. So, here if you see histone octamer, we have histone octamer. What are 4 proteins of histones? H2A, H2B, H3 and H4. Fine, right? 2 sets of this, H2A, H2B, H3, H4. 2 sets of this, fine, right? 2 sets of this will form one histone octamer. As such, the negatively charged DNA will coil around positively charged histone octamer. Likewise, one chain is connected to the other octamer. The DNA which is linking between 2 nucleosomes is addressed as linker DNA. Here, H1 histone proteins are present. Fine, right? So, one nucleosome bead, this is one nucleosome bead, this has 8 histone molecules and 146 base pairs of DNA. 146 base pairs along with linker DNA, it is 200 base pairs of DNA. 200 base pairs of DNA. Remember this is the complete content. Of course, first you have to present the diagram and then go for the theoretical explanation. Now, we will move on to nucleosome structure and its composition. Nucleosomes, its structure and composition. When chromatin is observed under electron microscope, it appears as what? Beads on string model. Okay, it appears as beads on string model. Each bead is a nucleosome here. Okay, each bead is a nucleosome, the fundamental unit of chromatin organization. This is the fundamental unit of chromatin organization. If you see the structure, each nucleosome is about 10 nanometers in diameter. It consists of a core particle of histone proteins. What does it contain, ma? Core particle of histone proteins which is wrapped around by DNA. Clear? Next, histone octamer. About histone octamer, this is made up of how many proteins? 8 proteins, 4 proteins, 2 sets of 4 proteins. H2A, H2B, H3, H4. Around this octamer, 146 base pairs of DNA are present. This is about 1.65 turns. Okay, this is about how much, ma? 1.65 turns, which are tightly wrapped. Next is linker DNA, the stretch of DNA, that is 50 base pairs connecting one nucleosome to the next. This is linker DNA. It helps in organizing the nucleosomes to higher order structure. What is the role of H1 histone octamer here? H1 protein in the octamer. What is the role of this? It lies outside the core and this locks DNA. This will lock the DNA at entry and exit points. This will stabilize the nucleosome structure. Okay, this will stabilize the nucleosome structure. Next is functions. What are the functions? Nucleosome compact the DNA, so it fits within the nucleus. Okay, it gets fit within the nucleus. They also regulate gene expression. They will also regulate gene expression by controlling DNA accessibility during transcription, replication and DNA repair. Fine, right? This is the function of nucleosome. Clear, right? First give a proper diagram, elaborate it properly, then move on to the functions of the nucleosome. Now, next question. Differentiate between rough endoplasmic reticulum and smooth endoplasmic reticulum. Very important question, quite a times asked, 2024 and 2018. Look into it carefully, Vita. First, here also I recommend to draw diagram only. How you will elaborate the diagram of endoplasmic reticulum? Extensions of nuclear envelope, the reticular structures over here, those which have ribosomes are addressed as RER, those which are just reticular extensions without ribosomes are addressed as smooth endoplasmic reticulum. Here we look into, ribosomes which are attached to the surface are known as what? Rough endoplasmic reticulum. They are also called as granular endoplasmic reticulum. Fine, right? This is also known as granular endoplasmic reticulum. This is useful for protein synthesis and secretion. This is prominent in protein secreting cells. Prominent in protein secreting cells like pancreatic cells. This helps in protein folding, for example glycosylation. Okay, smooth endoplasmic reticulum, no ribosomes at all. This is smooth in the appearance. This is also called as agranular endoplasmic reticulum. Clear, right? This is granular ER, this is agranular endoplasmic reticulum. Lipid synthesis will happen including phospholipids and steroids. Fine, right? This is prominent in lipid secreting cells, for example liver and adrenal cortex. This is involved in detoxification and glycogen metabolism. Fine, right? This is a clear differentiation between RER and SER. Next we move on to give the biochemical composition of plasma membrane, have lipid molecules arranged in the membrane, how they are arranged in the membrane. Here we go with biochemical composition of plasma membrane, for example lipid arrangement. First, take the fluid mosaic model only as a standard example. Give the structural description of the plasma membrane. First, elaborate the composition. It has what? Lipids which have hydrophobic head and hydrophilic tail. Hydrophobic tails and hydrophilic head of lipids. What type of lipids are present, like phospholipids, glycolipids and all you have to mention. And then proteins. Three types of proteins. Integral proteins, transmembrane proteins and peripheral proteins you have to mention. Carbohydrates in the form of glycolipids and glycoproteins. Then also mention about cholesterol. Here we go, overall composition. Plasma membrane is chemically made up of lipids. Okay, plasma membrane is chemically made up of lipids. It has 20 to 40% of lipids. 20 to 40% of lipids. Proteins 50 to 70% of proteins and carbohydrates 1 to 5% of carbohydrates. Hence, this is lipoproteinaceous nature. The accepted model is what? Fluid mosaic model, which is proposed by Singer and Nicholson in the year 1972. The proteins in the plasma membrane, if you see, we have three types of proteins: intrinsic, extrinsic. Intrinsic are integral proteins, extrinsic are peripheral proteins. Third type is transmembrane proteins, that also you can mention over here. Fine, right? Third is transmembrane proteins. Third will be transmembrane proteins. Integral or intrinsic proteins, they span the lipid bilayer or completely. Often they act as what? Channels, carriers and receptors. Peripheral or extrinsic proteins, they are loosely attached on the surface. They will provide mainly structural support and they will give the enzymatic activity. Next is transmembrane proteins. These transmembrane proteins, they will transpan either of the membranes. they will transpan either of the membrane side. Fine. Next, lipids in the plasma membrane. Mainly phospholipids with cholesterol and glycolipids. Lipids form a bilayer like hydrophilic heads which are polar. They will face outwards towards the aqueous membrane or aqueous environment. Hydrophobic tails which are non-polar, they are towards inner side, they are away from water. This arrangement will provide what? Selective permeability and stable barrier. Next is carbohydrates. They are present as glycoproteins, glycolipids on the outer surface. What is their main role? Like cell recognition, signaling, adhesion, most importantly cell to cell interactions. Next, dynamic nature. Lipids show lateral movement, making the membrane quasi-fluid nature. The fluidity will allow proteins to diffuse, interact and carry out the functions like transport and signaling. Functions you see ma, what are the functions of this? Functions include selective permeability, transport, cell communication and structural support. Just go through the functions clearly. If you are having any doubt, just drop a message. Here we go. The question is about types of chromosomes based on the position of centromere. Shapes of chromosomes, which is very important. Even in NEET and NIMSET we got many a times questions. Statement based, most importantly match the following type of questions. So if you see this question clearly, appeared in March 2020, 2018, 2016, May 2016 as well. Types of chromosomes you see ma, this is based on position of centromere. A chromosome with single centromere is addressed as what? Monocentric chromosome. Okay, with single centromere it is addressed as monocentric chromosome. In this monocentric chromosome we have these types. First we will see the metacentric chromosome, where two arms of the chromatid are of equal length, and this gives out V shape during anaphase chromosomal segregation. After centromere splitting, this will give V shaped chromosomes. Okay, V shaped metacentric, they will have equal length of chromatids. Next you see submetacentric and acrocentric chromosomes, where you will have one short arm of the chromatid, one long arm of the chromatid. As such, for submetacentric the chromosome shape will be L shape. For acrocentric, the chromosome shape will be I-shaped chromosome. Okay, and telocentric, here if you see, there is only one arm of the chromatid. Telocentric, there is only one arm of the chromatid. Clear, right? So here we have metacentric, submetacentric, acrocentric and telocentric. Depending on the position of centromere, chromosomes will be of four types. First is metacentric chromosome, where centromere is present in the middle, two arms will be there and it is of V shaped. Example is onion. Submetacentric, centromere is slightly off the center, one arm is long and the other is short, it is L shaped. Human chromosome 4. Example. Acrocentric, centromere is located close to one end, it gives J shape, one very long arm and one very short arm. If you can mention the names of long and short arm. Long arm is addressed as Q arm, short arm is addressed as P arm. If you mention that, that will be impressive. Next, telocentric chromosome. Yeah, telocentric chromosome. Centromeres is at the terminal end of the chromosome and it has only one arm. During anaphase, this appears as what shape? I shape. This is found in mice. This is the example. Next, last question of today's session. If you see, the question is to mention about two cell organelles, that is double membrane bound. What are the characteristics of the two organelles? State their functions by drawing neat labeled diagrams. One time it is asked. Two double membrane bound organelles, you can take anything, chloroplast, mitochondria or nucleus. Amongst these three, you can take anything. But here we are going to compare between two semi-autonomous organelles, that is chloroplast and mitochondria. Here you see chloroplast and mitochondria. Of course, both are double membrane bound. Intermembrane space is periplastidial space, it is perimitochondrial space. This is found only in green plants, this is found in both plants and animals. And here if you see, inner membrane forms what? Flattened sacs. Those sacs are called as thylakoids and they are folded into grana interconnected structures. Here mitochondria, inner membrane is folded to some projections called as cristae, okay, inner mitochondrial membrane. What does this contain? Chlorophylls and carotenoids. This contains enzymes which are required for ETS, electron transport system. Here photosynthesis, both light and dark reactions. This chloroplast is addressed as kitchen of the cell. Here mitochondria is addressed for cellular respiration, Krebs cycle in matrix, electron transport in inner mitochondrial membrane, oxidation of food and ATP production. This serves as energy currency of the cell. These are semi-duplicative, semi-autonomous or self-duplicating organelles. Mitochondria is addressed as what? Powerhouse of the cell. Fine, right? Here we will finish off what? The comparison or comparative analysis between chloroplast and mitochondria. With this, we have dealt with short answer type of questions from two chapters. One is sexual reproduction of flowering plants and second will be cell the unit of life. In our next class, we will also address the questions on biomolecules and cell cycle and cell division, where we will also solve from some numerical based questions as well. So, that is all for today's session. In our next class, we will come up with the better questions linking up the preparation with MCET and NEET. Okay? So, stay tuned and thank you.